Perfect. Thank you so much. So thank you for this kind introduction and thank you for this invitation to present. It's an absolute honor. My research program centers on understanding how microbial members of the microbiota, in particular bacteria, impact the tumor microenvironment. And if we count these microbes that reside on our bodies, it's estimated that we have about 39 trillion microbial cells. But these microbes outnumber our 30 trillion human cells. And it's also estimated that about 95% of these microbes reside within our gastrointestinal tract, largely in the large intestine. And it's here where these microbes set the stage for dynamic interactions in human health and disease processes that includes cancer. Because when cancers arise along the gastrointestinal tract, all the way from the oral cavity to the colorectal, we and others have assessed microbial communities within the tumor tissue and compared them to adjacent normal tissue. And what we see are distinct bacterial taxa that are enriched in the tumor tissue compared to the normal tissue. And one such organism is a bacterium called Fusobacterium nucleatum. And I'm going to talk a lot about this microbe today. And this is a microbe that we see consistently enriched in tumor tissues along the gastrointestinal tract compared to adjacent normal tissues. And why this organism is interesting and important is because there has been large-scale epidemiological studies that have shown when a patient's tumor harbors a high load of this microbe, the patients have a poor prognosis. There's an increased risk for the cancer to recur, and there's also an increased risk for death. We don't fully understand why that is. And I began working on this organism over a decade ago when I joined Matthew Myerson's group at the Dana-Farber Cancer Institute in Boston. And one of the first questions I asked was, given that we know this microbe, Fusobacterium nucleatum, is enriched in colorectal tumors, what happens when those colon cancers migrate or travel to a distant site that happens in metastases? So what happens with the association of the cancer and the bacteria when the tumor travels to the liver in metastases? And what we saw was that about 20% of the liver metastases harbored Fusobacterium. Again, these are liver metastases. The primary tumor is in the colon. And we saw that all of these positive liver mets had a paired positive primary colorectal tumor. So telling us that if the liver metastases was positive for Fusobacterium, the primary tumor was also positive. So suggesting this persistence of the bacterium as it travels with the cancer to a distant site in the body. We also looked at a different patient cohort, again, where we had a paired primary tumor and a corresponding liver metastases. And here, these were snap frozen tumor tissues. So I could carry out microbial culture on these tumors. And I was able to culture Fusobacterium from both the paired primary tumor and the distant site metastases from two patients, sequence their genomes, and confirm that the exact same strain of Fusobacterium colonized both the primary tumor and the distant site metastases, despite these tissues being resected up to 2 years apart. So really demonstrating this persistence of the microbe with the cancer. We also carried out microbiome analysis in these tumors because Fusobacterium is never alone. It's never just a single organism in these tumors. There's a community in these colorectal tumors. And what we saw was that in addition to Fusobacterium persisting in the tumor, we also saw other anaerobic microbes that were also present in the primary tumor persisting in the metastatic disease. These included gut microbes like Bacteroides species, but they included quite a lot of oral pathogens. So in addition to Fusobacterium nucleatum, Fusobacterium necroform, we saw Treponema species, we saw Prevotella species, we saw Senomonas species. So many oral pathogens persisting with Fusobacterium in the cancer as it spreads to these distant site metastases in the liver. So what this tells us is that there's this close association between the microbe and the particular tumor microenvironment. And because we had this model system where we had the persistence of the bacteria in the tumor, we could implant it into a mouse xenograft. And we could ask, what would happen if we treated these tumors that harbored these anaerobic bacteria with an antibiotic that targeted anaerobes? And here we chose metronidazole. And what we saw was that antibiotic treatment of these patient tumors in mouse models led to a significant reduction in tumor growth. So this was really a proof of concept that if you could target members of the intratumoral microbiota, it might help in the treatment of a subset of colorectal cancer. But as you know, metronidazole is a relatively broad spectrum antimicrobial, and it targets a range of different anaerobic bacteria. So when I started my own group, one of the many projects we had ongoing was trying to identify a Fusobacterium growth inhibitor, and a very narrow spectrum inhibitor, so that it would specifically target Fusobacterium and not other members of the gut microbiota. And to do this, we initially began a small molecule screen. And here I'm showing you the layout. And this was work led by LaCourse, who was a postdoc in my group at the time. And this was a very simple approach where we had a range of small molecules printed on plates in duplicate. And they were at a specific 32 micromolar concentration. And we grew Fusobacterium in clinical isolate. And we seeded Fusobacterium into these wells and grew for 48 hours and measured growth through ATP. And this was a luminescent assay. And when we initially began working with this, setting up our conditions, we used a pilot study. And what was interesting from the pilot study is that we had a serendipitous finding that in addition to our control metronidazole inhibiting Fusobacterium growth, what we saw was that the routine chemotherapeutic that's used to treat patients with colorectal cancer, along with its prodrugs Tegafur and a range of other prodrugs to 5-fluorouracil, were potent inhibitors of this bacterium's growth. So this microbe, which has been reproducibly shown to be enriched in human colon cancers, is inhibited by the routine chemotherapeutic drug that's used to treat patients with colorectal cancer. And this was interesting because it raised the possibility that patients are responding to this chemotherapeutic in part due to its antimicrobial activity against Fusobacterium nucleatum. And we confirmed this growth inhibition with a range of clinical isolates of Fusobacterium nucleatum. And importantly, its growth, Fusobacterium nucleatum's growth, was inhibited within the biologically significant range of this chemo drug that patients with colorectal cancer would receive. We then looked to see if this chemo drug had broad spectrum antimicrobial activity. So we had a range of different microbial isolates from human colorectal tumors, including Bacteroides fragilis, E. coli, we had B. breve, we had Parvovirus micra. And what we saw was that it did not have broad spectrum antimicrobial activity. So it was inhibiting Fusobacterium nucleatum, but not these other microbes. But when we looked through mass spectrometry at 5-fluorouracil levels after exposure to these other gut microbes, we saw that Escherichia coli was metabolizing or modifying this cancer drug, whereby at 24 hours we could no longer detect it in the media. And this actually is in line with other studies. Peter Turnbrock's group also had a paper demonstrating certain microbes could metabolize or modify this routine chemotherapeutic that's used to treat patients with colorectal cancer. But what was interesting was that when we looked at these other microbes that in exposure with 5-fluorouracil, what we saw was that when E. coli was co-cultured with 5-fluorouracil, 5-fluorouracil could no longer kill Fusobacterium nucleatum. So it was rescuing Fusobacterium nucleatum. And what you can see here in these bar plots is Bacteroides fragilis had no impact. So the spent media had no impact on rescuing Fusobacterium. Again, as we can see with our mass spectrometry analysis, B. fragilis was not metabolizing 5-fluorouracil. But the E. coli strains that were co-cultured with 5-fluorouracil, its spent media now no longer inhibited Fusobacterium nucleatum's growth. So there was no 5-FU now detected after 24 hours in co-culture. So we also did this experiment with cancer epithelial cells. And again, we saw that these RKO colon cancer epithelial cells could be rescued from 5-FU toxicity if E. coli was co-cultured with 5-fluorouracil. Again, this is spent media that we are exposing Fusobacterium nucleatum to and the colon cancer cells. So what this tells us is that particular members of the microbiota or the intratumoral microbiota have the ability to rescue both Fusobacterium nucleatum and cancer epithelial cells from the chemodrug toxicity. And when we looked back at our patient specimens, we had 6 colon cancer tumors from unique patients. We generated ex vivo CRC communities, so these ex vivo microbiota communities, and carried out a range of analyses. And in particular, we looked at mass spectrometry and asked, could these members of the microbiota that came directly from a patient's tumor metabolize this cancer treatment drug? And what we saw was that about 50% of the patients harbored a microbiota that could metabolize this chemo drug within 48 hours. So within 2 days, we could not detect this routine cancer treatment drug. So what this tells us is that there's an interplay between these microbes that are residing within the patient's tumor and the cancer drugs that they're receiving. These microbes are metabolically active, and they can interact with these cancer drugs, metabolizing them, modifying them into an active form that can rescue the cancer epithelial cells. But what we're also seeing is that many of these cancer treatment drugs are anti-metabolites, and they're having anti-metabolic activities on some of these intratumoral microbes like Fusobacterium nucleatum. But depending upon what other microbes are present with Fusobacterium nucleatum, they can be rescued by this 5-FU toxicity. So this really demonstrates complex interplay between the drugs and the microbes within the tumors, and it's something that's largely overlooked in cancer research, and that can greatly impact patients' response to treatments. And when I've been talking about Fusobacterium nucleatum up until now, I've been referring to it as the sensu lato species. But within Fusobacterium nucleatum, it's an incredibly heterogeneous species. And there are many subspecies within this larger species. And one of the questions we wanted to ask within my group was, are there specific strains or specific subset of Fusobacterium nucleatum that is associated with human colorectal cancers? And the reason we asked this is because Fusobacterium nucleatum, as you may know, is typically found in the oral cavity. It's noted to be a periodontal endodontic pathogen associating with oral health and disease. It's rarely found below the oral cavity in non-cancer individuals or in patients that do not have inflammatory diseases in their gastrointestinal tract. Yet in colorectal cancer, this microbe can dominate the tumor microenvironment. So we hypothesized that the strains of Fusobacterium nucleatum that were capable of getting to the colorectal tumor and colonizing the colorectal tumor had unique genetic attributes. And to assess this, we carried out a range of traditional microbial culture approaches. And this work was led by Martha Zepeda Riviera, who was a postdoc in Chris Johnson's lab and my lab at the time. And then she's now a staff scientist within our group and moving to instructor. So to start this analysis, we obtained 59 patient, actually more than 59, this is where we cultured from, we were at close to 120 patient colorectal tumors. These were treatment naive primary colorectal tumors, and we carried out targeted Fusobacterium culture on these tumors, from which we isolated 65 unique strains. We also collaborated with Dr. Junkun Ki at the KCOM, where we also obtained oral isolates. So these were isolates from patients that were presumed healthy that did not have cancer. And we wanted to compare the Fusobacterium strains that were colonizing tumors to those that were routinely present in the oral cavity of non-cancer patients. So we compared 65 strains from the CRC tumors to 81 strains isolated from the oral cavity of non-cancer individuals, carried out SMART sequencing through PacBio, and we generated a collection of genomes and methylomes. So we initially identified Fusobacterium strains in these patients through 16S sequencing, and just through sequencing the full length of the 16S, which is incredibly common when looking at Fusobacterium nucleatum and trying to type Fusobacterium as a preliminary step. And what we saw that was largely Fusobacterium nucleatum, that was isolated from both the oral cavity and from the CRC tumor. So we focused in particular on nucleatum at the species level. And as I mentioned earlier, within Fusobacterium nucleatum, it's an incredibly heterogeneous species. There are 4 known subspecies. This is nucleatum subspecies nucleatum, nucleatum subspecies polymorphum, nucleatum subspecies animalis, and nucleatum subspecies vincentii. And all 4 of these are present in the oral cavity. Yet we do not know their prevalence in the colorectal tumors. So again, as I mentioned, we had 16S data where there was preliminary typing. And with this preliminary typing, what it appeared was that it was only this subspecies animalis that was associated with these colorectal tumors. And it was actually the opposite for subspecies nucleatum, where it was largely in the oral cavity and very rarely in the colorectal tumors. So given that we saw this enrichment of subspecies animalis in the colorectal tumors, we wanted to ask if there were any enrichment for known virulence factors or canonical virulence factors that are associated with this particular microbe, Fusobacterium nucleatum. So we looked at these virulence factors, BAD-A, we looked at FAP2, we looked at RAD-D, these adhesins, and fusolysins, looking to see if there was an increase in copy number or an increase in prevalence. And actually what we saw was the opposite. So when we looked at this group that was typed as animalis through the 16S, what we saw was that within the group, there was actually less, there was a subset that had less of these known virulence factors. And as you can see here, it appears that this group that has less is separating from this group on the left. So there's a group on the left that has more of the virulence factors and a group on the right that does not. And here you can see a niche isolation based upon the color coding. So we've got this kind of teal color for oral, and we've got this green or this orange color for colorectal. And you can start to see a trend based upon this presence of virulence factors and the niche association. So when we looked at this group further, we looked at it through K-SNP analysis, we looked at it through average nucleotide identity, and we looked at it through GigMap, looking at protein coding genes. And as you can see, these are not one single group. There is a bifurcation from a common ancestor where we have this group on the left and we have a separate clade on the right. So there is this two distinct clades within what was being typed as Fusobacterium nucleatum subspecies animalis, but they really are genetically distinct, as we can see through K-SNP, ANI, and we can see through protein coding genes. Again, bifurcating from a common ancestor into 2 distinct clades. And when we looked at their niche localization, what we saw was that both of these clades were present in the oral cavity. There is no significant difference in their prevalence. Yet it was only this clade 2, so this purple clade, so again, if we look back at the color scheme, we're calling this group here on the left clade 2, and this green group as clade 1. It was only this clade 2 that was significantly enriched in the colorectal tumor niche. And why this is interesting is because both of these groups were being classed through 16S as subspecies animalis, but through whole genome sequencing, they're clearly distinct, genomically distinct groups. So we have these 2 clades that are the nearest neighbor to one another. So they're phylogenetically each other's nearest neighbor. One group is enriched in the colorectal tumor, and both of these groups are found in the oral cavity. So these are a perfect group for comparative analyses to try and identify what are the factors that facilitate this clade 2 group's enrichment in colorectal tumors. And we do a range of these analyses in our published paper, and I don't have time to go into details of those now, but it's largely to identify factors that facilitate niche colonization of the tumor. And we've identified ethanol amine utilization, phosphodiol utilization, so many processes that are associated with enteric pathogens. We also carried out analyses in a mouse model where we had our vehicle control in arm 1. Arm 2 was this clade 1 treated group through oral gavage, and then arm 3 was this clade 2 treated group through oral gavage. And it was only this clade 2 treated group that increased intestinal adenomas. So again, supporting what we're actually seeing in our patient data, that not all Fusobacterium nucleatum behave the same, and that it's largely this clade 2 group that is enriched in the colorectal tumors. But all of this correlation was carried out through culture. We initially discovered it through culturing whole genome sequencing. So we wanted to ask, was this also observed through culture-independent approaches? And here I'm showing you patient colorectal tumors, and this is V3, V4, 16S sequencing. And here we looked at 62 patients, and we have subset Fusobacterium abundance based upon the different species. And here you can see in the key what these are. The tumor is orange and the paired normal tissue is black. And what we can see is that similar to previous results worldwide, it's this F nucleatum at the species level that's significantly enriched in the tumor compared to the paired adjacent normal tissue. When we break the Fusobacterium nucleatum group into its subspecies, and in particular into this Fusobacterium nucleatum clade 1 and clade 2 within Animalis versus all other subspecies, we can see that this enrichment is being driven by this clade 2 group. And we collaborated with Nicola Zagato's group to also look at the prevalence of these two clades within the stools of individuals that have cancer compared to healthy individuals. And when we account for this overlapping group, it's actually only this clade 2 that's significantly enriched in colorectal tumor stool from patients with colorectal tumor compared to healthy controls. So what this is demonstrating is that, again, within Fusobacterium nucleatum sensu lato, there is an incredibly heterogeneous population, and that it's only this specific Fusobacterium nucleatum subspecies Animalis clade 2 that's enriched in the tumors. And this clade 2 contains the type stream, so will become Fusobacterium animalis sensu stricto. And this should be the focus of future studies that are trying to gain mechanistic information on Fusobacterium nucleatum's role in colorectal cancer and the design of therapeutics that target Fusobacterium nucleatum in the context of colorectal cancer. And that brings me to the next part of my talk, which centers on the tumor microenvironment. Because when we think about cancer, we'll often think about this ball or this mass of cells. This mass of cells is incredibly heterogeneous itself. And when we think about cancer, we might think about the cancer cell. But the cancer cell, what are the driver mutations? Can we target them for treatment? But within a patient's tumor, these cancer cells are surrounded by a range of different cells. We have infiltrating immune cells, we have fibroblasts, we have endothelial cells. And we now know in a subset of cancers, especially those along the gastrointestinal tract, we have this infiltrating microbial component. And each of these components of the tumor microenvironment are capable of interacting with one another to impact cancer progression and also to impact patients' response to treatments, whether that's chemotherapies or whether that's immunotherapies. But we still have very little information on how the microbial component is impacting the human component of these cancers. And the reason why it's important to understand this microbial component is because the microbial component is malleable. We can modify it, we can manipulate it. So if we can understand how it's impacting the human side, it opens up new avenues for therapeutic development and new treatment approaches. So within this context, my group set out to ask and answer several unknown questions relating to microbes that are infiltrating human tumors. So firstly, where do these microbes physically reside when they enter the tumor? What are the human cells that they're interacting with? And importantly, what are the consequences of these host-microbial interactions within patient tumors? And this work that I'm going to talk about next was led by a wonderful postdoc in my group, Galeano Nino, who is now faculty at El Paso, with support from LaCourse and Hungary Wu. And the first question we set out to ask and answer is, what is the spatial distribution of these intratumoral microbes within patient specimens? And this is just a background slide because this is to demonstrate that traditionally, in tissue microbiome analysis of these patient tumors, what we would do is we would obtain a patient's tumor, and it's a piece of tissue, and we would just homogenize it. So it would be bulk tissue analysis. And because most of the nucleic acids within this type of specimen are going to be host, they're going to be human, what we traditionally do is 16S sequencing. Rather than metagenomic analysis, because the microbial component is minor compared to the human component. And when we're carrying out the microbiome analysis, we're interested in their composition, so who's there, what proportion, and we're interested in diversity. What's often overlooked is the microbial load, so how much of a particular microbe is present in relation to the human cells. And this is important because it varies drastically depending upon the cancer type. So cancer types that are outside of the gastrointestinal tract will have a far lower microbial load, and those cancer types are more susceptible to issues relating to contamination and skew relating to contamination. So load is incredibly important, and it varies drastically depending upon the cancer type that is assessed. So when we started this project, we wanted to carry out a simple reproducibility study where we would take one of these patients' tumors and we dissected it into 4 random but equal parts, and we carried out microbiome analysis through 16S. And we asked, does it give us the same result? Depending, depending upon what piece we assessed from the patient's tumor, do we get the same result? And what we saw was no, we did not. There can be incredibly different results depending on the piece of the patient's tumor that we assessed. So for example, if you look at patient colorectal cancer 7, and we look at Fusobacterium's relative abundance, we can see that it ranges all the way from about 5% relative abundance to over 80% relative abundance, depending on what piece of the tumor tissue that you've taken. So what this tells us is that there's regional variation in microbial distribution within a patient's tumor. And here you can see RNA-scope imaging. So this is imaging of patient tumor tissue. So here I'm showing you 2 cancer types at the extremes of the gastrointestinal tract. We have this oral squamous cell carcinoma of the tongue, and we have a colorectal tumor. And with this RNA-scope approach, we have probes that target eubacteria, and that's seen in this cyan green color. And we have probes that specifically target Fusobacterium nucleatum, that is this dark red color. So I hope what you can appreciate is this signal is not evenly dispersed across the tissue. We have this focal distribution of bacteria in specific regions of the tumor tissue. So what this tells us is that bacteria are not evenly present across the tumor, that they're forming these microniches. And this goes back to the tumor being heterogeneous. The tumor itself is a microenvironment, and in different pockets of the tumor, you have hypoxic regions, you have normoxic regions. So the environment is very different depending upon the region. And in cancer research, there's a whole area, a whole field of study centering on the tumor microenvironment and how it's this diverse ecosystem across the tumor tissue, and bacteria are even representing that. So we have specific regions where these microbes will colonize, reflective of that ecosystem where they're localizing. So to gain more information on this, we did adapt, and it was an emerging technology at the time. Now it's totally outdated, but it was 10x Visium spatial transcriptomics. And I won't go into too much detail here because I am coming close to time. But what this allowed us to do was to take advantage of sequencing reads that were generated from patient specimens that were placed on slides that have capture spots. And each of these sequencing reads had a unique molecular identifier, which was a proxy for transcriptional load, along with a spatial barcode, allowing us to map the reads back to the patient specimens. So we used GATK-PAT-seq to run the host-generated sequencing reads through GAT-Patsy, where we took unmapped reads and aligned them to a highly curated microbial database. What we showed through this approach, and also having RNAseq on a sequential section to validate this, was that indeed within a patient's tumor specimen, these microbes are localized to specific regions of the tumor tissue. They're not universally distributed across the tumor. And we can identify what taxa are present in situ within the patient tumor specimens. And here I want to just confirm that the RNAseq probe was actually targeting a different region of the 16S and the 23S. So these are two independent approaches validating each other, again confirming this localization of bacteria to very specific regions in both these oral cancers and colorectal cancers. So here I'm showing you a patient's colorectal tumor, and this is a video where we have Fusobacterium nucleatum stained red, and we have our eubacterial probe as a cyan color. And what this is showing is that there's regions of the patient's tumor where we have dense infiltration of bacteria, regions where bacterial cells are outnumbering human cells. And when we zoom out, there's regions of the tumor where we have absolutely no microbial signal. And what we're interested in understanding is if there is something unique about the regions of the tumor where these microbes are localized that allow them to colonize, that allow them to persist. And to understand this, we applied another spatial technology. This time we used nanostrings digital spatial profiling. And I won't go into details on the approach, but we did use an antibody-based approach here, where we had 77 antibody panel that targeted proteins relating to oncology and immunotherapy. And our approach here was 2 sequential sections. So these are 4-micron sections of patients' tumors. We looked at oral cancers and colorectal cancers. We carried out RNAseq to identify the regions of the tumor that harbored bacteria. And then we used this digital spatial profiling to select regions that were bacteria positive or bacteria negative, and we could compare them to ask, is there a difference in the regions where bacteria are localized? And when we did this analysis on these 2 different cancer types, we saw similar results. So here I'm showing you volcano plots. On the left, these are proteins that we have shown to be enriched in the regions where bacteria are present. And on the right, these are proteins that are depleted in the bacteria-positive regions, enriched in the bacteria-negative regions. So what we saw in these 2 cancer types is that these microbes were largely localized to regions of the tumor that were immunosuppressive. And in the context of cancer, an immunosuppressive environment is not favorable because you want your immune system to recognize the cancer cells and kill them. So if it's an immunosuppressive environment, the cancer cells can persist. What we saw was that there was an infiltration of myeloid cells, such as macrophage, neutrophils, characterized by CD66B, arginase 1. We saw there was an increase in immune checkpoint protein, CTLA4, LAG3, PD-1, again, suppressing the ability of T cells to kill the cancer cells. We also saw that there was an exclusion of T cells in these regions of the tumor where there were bacteria. So we saw less CD8, less CD3, less CD4. So again, excluding these T cells. And we saw also an upregulation of MAP kinases that are important in cancer cell growth progression, such as phospho-ERK, phospho-P50, or P38. But the key question here was whether these microbes were actively contributing to shaping this tumor microenvironment, or were they just responding to an environment that already exists and colonizing an environment that already existed. And to test this, we had to go back to functional studies in the lab. And here I'm showing you colon cancer spheroids that have been co-cultured with neutrophils. The neutrophils are purple. On the left, there is no Fusobacterium, and on the right, we have added Fusobacterium. And in the presence of Fusobacterium, these neutrophils migrate into the center of the cancer cell spheroid. They form these large clusters and become trapped. Very similar to what we are seeing in the patient's tumors, demonstrating that these microbes infiltrating the tumor can actively shape their infected tumor niche. And we also saw the neutrophils upregulate phospho-P38 in a dose-dependent manner in the presence of Fusobacterium nucleatum. But what about epithelial cells? So here I'm showing you colon cancer epithelial cells. On the left, no Fusobacterium, and on the right, we can see Fusobacterium as pink being added. So without Fusobacterium, the colon cancer epithelial cell grows en masse as a spheroid. But when Fusobacterium is added, the colon cancer epithelial cells migrate at the single cell levels. We see these individual cancer cells migrating off the main spheroid, and they're bringing Fusobacterium with them. And when we carry out analyses on gene expression of these cells that are migrating with Fusobacterium, we see upregulation of pathways that are associated with metastases, extracellular matrix remodeling. So again, suggesting that this microbe is helping the cancer progress. And we also see upregulation of MAP kinases in a dose-dependent manner in the presence of Fusobacterium. Again, resembling what we saw in our patient specimens, that these microbes are actively changing the environment that they're infecting within the tumor. And here I'm showing you confocal microscopy. You've just seen single cells migrating off the tumor with bacteria associated. And here you can see this clearer through colon cancer epithelial cells that have been co-cultured with Fusobacterium nucleatum. And this is the center of the Z-sac. So we've cut into the cell. And here on the right, you can see the red Fusobacterium. The actin is green on the cell and DNA of the cell is blue. And this particular cancer cell is undergoing metaphase. The nuclear envelope is broken down and we have bacteria right next to the chromosomes of the human cell. What happens next? We do not know. So this is where we developed a single cell sequencing approach, and I won't go into details because my time is almost up, but here this approach is called INVADEseq. We published a protocol on this, a very detailed protocol, so happy to answer any questions on that through email if you have more questions. But what this approach allowed us to do was identify what proportion of human cells harbor bacteria, what are the identity of the bacteria and the human cells, and understand how these microbes alter the transcriptional profiles of the cells that they are associated with. And again, these are single cells here on the left that have come directly from a patient's tumor, and we can identify adherent and invasive bacteria in the cells directly from the tumor. And in the last minute or so, I'm just going to show you some of our single cell sequencing data from these patients' tumors. So here we have oral cancers of the tongue. So these are oral squamous cell carcinoma. We have 7 patients. We generated close to 42,000 single cells. These are human single cells. We can annotate what their cell type is based upon their gene expression. And this is a UMAP plot of what the single cells are. So we have our epithelial cells, we have a range of immune cells. And with our INVADEseq approach, we can now use the 10x barcode to map our microbial reads back to the individual cells that they've come from, with the premise that a single cell, a gem within the 10x, the microbe and the single cell, the whole single cell will share the same 10x barcode. And we have a unique molecular identifier, which is a proxy for transcriptional load. So here you can see the bacterial taxa that we identified in these oral cancers associated with single cells. We can see that they're largely in a subset of macrophage, which makes sense, and also in a subset of epithelial cells. And when we analyzed these epithelial cells, we actually confirmed that they were cancer epithelial cells, not just normal epithelial cells. They're aneuploid, which is a chromosomal abnormality that we see in cancer. But with this approach, we can now look at any of these taxa that we're interested in. So for example, I'm just showing you Fusobacterium. So in these oral cancers, there was largely Fusobacterium and Treponema species that were associated with the single cells that were cell adherent and cell invasive. And when we look at Fusobacterium, again, we can see that it's in the subset of macrophage and in the subset of epithelial cells. But we can now say, how does a single cell that's a cancer cell, differ from a single cell that's a cancer cell that harbors Fusobacterium? So we can compare the cell types directly based upon the presence or absence of cell-associated bacteria. What we see is that the macrophage, they don't care what bacteria are present, they behave in a certain way, but the cancer epithelial cells have very distinct functions depending upon whether it's Fusobacterium or Treponema or Campylobacter that are present. So this will allow us to understand how these microbes are directly impacting the cells that they are associating with. And I'm going to finish there, but what this work tells us is that these microbes are not evenly distributed within a patient's tumor. Instead, they localize to distinct niches within the tumors that we call bacterial colonized microniches. And they're not innocent bystanders. They are changing the function of both cancer epithelial cells and immune cells to support cancer progression. And with that, I'd like to firstly thank patients and their families who have participated in this research, my close collaborator, Dr. Chris Johnson in his lab, along with Jorge Galeano Nino, Martha Zepeda Riviera, and other members of my group that helped with this work and drove this work forward, my funding, and thank you for listening. And with that, I would be happy to answer any questions.